Kimberly Narain, and my descriptive disease project is on malaria. Some purpose and objectives of this little PowerPoint is that we're going to learn what malaria it is and how it affects us. We're going to determine the causes of malaria in different locations around the globe, and we're going to examine how malaria is transmitted and examine trends and data about malaria infection and death. So just a little table of contents for this lecture. We're going to look at what is malaria, some signs and symptoms, causes and origins, spread and infection, treatment of disease, and then we'll have references at the end. So, what is malaria? Well, malaria is an acute febrile disease, which basically means you'll see a rapid onset of fever and other symptoms. Malaria is a prime example of a parasite-host relationship, but more specifically a parasite-host vector relationship, but we'll talk more about that later. A parasitic relationship is when a parasite lives off of another organism called the host, potentially harming it and possibly causing death. Parasite lives on or in the body of the host, so you can think of things like tapeworms, fleas. The specific parasite that causes malaria is called the Plasmodium falciparum parasite, and we'll also talk about that one later. A more common piece of info that a lot of people know is that malaria is spread through bites from infected mosquitoes, and that's a large part of the reason why it's such a health threat today. Since it's spread by mosquitoes, we can see why it's so common in tropical countries, because this is where mosquito populations are very high. Also, a little fun fact, the word malaria is Italian for bad air, which is kind of funny considering that you get malaria from mosquitoes, which are usually in the air, and they're usually because of hot temperatures. So, some signs and symptoms of malaria. It's important to keep in mind that a lot of the signs and symptoms exist on a very large scale. Some people may experience some things that kill them, whereas some people don't experience that symptom at all. Or a symptom could come on very slowly for one person and very rapidly for another person. The symptoms that we see are extremely non-specific, and they can be really easily confused with other viral infections. So, for example, you can see headache, nausea, diarrhea, joint and muscle pain, which are also common with viral infections. So, again, they exist on a large scale, so some people may experience different things at different levels than other people. One of the unique symptoms that you don't really see in other diseases is intermittent spikes in fever. So every few days, someone with malaria might get a high fever that dies down, only to come back up in another few days, but this also varies depending on the species of the malaria parasite. And because of the variability, the only sure diagnosis of malaria can be determined from a blood test or a microscopy that for the presence of the parasite in your blood. So, some causes and origins. Today's malaria parasite is likely to come from a genus that originated in Africa about 30 million years ago, and since then it has spread worldwide. Because of how widespread and how long the population of the parasite has been alive, the disease has several geographic and biological factors that affect its prevalence. This is why we see a lot of variability in different symptoms. A large reason that it's such a big health conflict today is because of the complicated relationship between the host parasite vector and the environment. The host is the human, the vector is the infected mosquito, and the parasite is the Plasmodium parasite. The parasite requires both the mosquitoes and humans to exist and to reproduce. The parasite uses the mosquito as kind of like a vessel to enter the bloodstream of the human while the mosquito is feeding on the human's blood. Because of the presence of a vector, it's hard to control the parasite because you can't directly prevent it from infecting humans. The only option is really restricting the vector itself. So, aka like mosquito nests, mosquito repellents are like good ways to prevent infection. But of course, everywhere on Earth, there are like different genetic groups and different environmental conditions that change the method and the frequency in which it's spread. So, since there's so much variability again, it's really important to consider in a global context. So, it's very difficult to effectively treat, diagnose, and track, which makes it much easier for it to spread. Some social conditions, yeah. So, almost 2/3 of malaria cases are in sub-Saharan Africa, largely because the resources needed to test, diagnose, prevent, and treat malaria are harder to come across in these areas. These countries are often poorer and can't afford these resources, so cases often like skyrocket. This is also true for other poorer tropical countries. So here we have a pie chart of all the countries that had the most malaria cases in 2020. In previous years, the trends were pretty similar. We can see that the largest percentage of the graph is from Nigeria, an African country, and it's 27%, which is a lot. A majority of the other countries with leading malaria rates are also in sub-Saharan Africa, as I mentioned in the other slide. And it's really unfortunate because African countries kind of have the worst of both worlds, with the ideal weather conditions for malaria to spread, and they're also typically lower income areas that can't afford prevention, tracking, testing, and treatment, making cases really difficult to keep at lower numbers. It usually spreads very easily, and when you don't have the ability to control the spread or treat the infected people, more and more people get infected very quickly. Just to take a deeper look at how drastic the problem of malaria is in Africa, here we have a graph showing the estimated death rate of malaria worldwide during 2015 and 2020. An extremely large percentage of the deaths in both 2015 and 2020 were in Africa, around 60% for both years, raising slightly. And if you really consider this statistic, it's actually shocking. So, like, almost 2/3 of global disease occur in one area of the world. And it really shows how, like, out of hand the situation is and how many resources that would be needed to reverse this effect. Even over time, like, it's still, like, a large percentage and it's only getting higher. The cases and deaths would be nearly cut in half if malaria didn't occur as heavily as it does in Africa. So, as for spread and prevention, malaria exists in a life cycle. So, a mosquito will infect a human, and the infected human infects a mosquito, and so forth. While that sounds simple, the cycle is very complicated and very intricate. There are 3 different cycles within malaria's life cycle: the exoerythrocytic cycle, the erythrocytic cycle, and the sporogonic cycle. I'm going to go into a little more details on these in the next slide, but I also want to mention that malaria prevention is largely based on controlling the spread of malaria. As I mentioned before, malaria uses a vector, making it hard to directly attack or limit the parasite, so we rely on preventing the vector from infecting the host. However, in order to control the vector from infecting the host, we really need to understand how the transmission is occurring. The timing, the methods, the biological factors all play a role into how we can effectively prevent infection. So this is the full malaria infection cycle, and it starts with the exoerythrocytic cycle. So in this cycle, the infected female mosquito bites a human and feeds on the blood. During this, the sporocytes from the female mosquito's saliva are injected into the human's bloodstream. These sporocytes migrate to liver cells in a very short period of time, actually. I think it's 30 minutes. And once they're in the liver cell, the sporocytes develop into infected liver cells called joints, which are made up of thousands of merocytes. And several strains of malaria, such as P. vivax and P. ovale, have dormant forms, and they can stay in the squints and relapse up to 5 years later. So now in the erythrocytic cycle, which happens about 5 to 21 days after the exoerythrocytic cycle, the mature squints will break in the bloodstream, and they can either, each of the mesozoites can either go back and infect another liver cell, or they can infect a red blood cell. The merozoites then, they can reproduce and form trophocytes. When trophocytes mature, they're called erythrocytic squints. When the mature erythrocytic squints break, they can infect other blood cells, making another cycle, and this can happen over varying periods of time, the release of the merozoites, and over this varied release of time is what causes intermittent spikes in fever, which is a unique feature of malaria. Also, when trophocytes are maturing, at times they can turn into gameocytes, which kickstarts the sporogonic cycle, where these gameocytes develop into male and female gametes. And when a mosquito feeds on another, an infected human's blood, they fertilize in the mosquito and form a zygote. And these zygotes can turn into eukenites. And when eukenites invade the gut of the mosquito, they form ooists. After 2 to 3 weeks, the ooist is mature, and it can break, releasing sporocytes into the salivary glands of the mosquito, making it infectious. And when this mosquito bites a human, the cycle repeats at the exoerythrocytic cycle again. It's a very complex, and timing is clearly a very big factor because of all of the processes that are taking place. So, treatment. A large part of treating malaria is diagnosing it correctly. Currently, the most effectively diagnosed from blood smears, where the presence of the parasite can be seen within the blood and the species can also be identified. And both of these pieces of information are vital in treating and managing an infection because the disease differs by species of parasite and it needs to be treated differently. These tests, however, require exact and rigorous methodologies, which are hard to attain in poorer countries of the world. However, there are different types of tests, such as polymerase chain reaction assays and rapid diagnostic tests. However, assays are often expensive and complicated. Rapid diagnostic tests, on the other hand, are they can quickly and accurately detect malaria and antigen proteins. So in poorer countries, they're a pretty decent alternative that can be widely available. However, they don't really inform the species of the parasite, and the test itself is sensitive to temperature, so it's hard to keep stored. One important factor that prevents a serious infection of malaria is the sickle cell trait. With this trait, one's red blood cells are sickled, they're disformed, making it difficult for the parasites of the Plasmodium falciparum malaria to develop or infect the blood cells. While having sickle cell anemia has mostly negative effects in some parts of the world, having it is really beneficial in locations with high rates of anemia. With this trait, if you're infected with malaria, there are fewer blood cells that they can successfully invade. This reduces the symptoms and illness associated with malaria. People residing in regions with large amounts of malaria cases may also acquire immunity through natural exposure. After multiple infections over a large amount of time, human immune systems can reduce the effects of infection. Lastly, other common treatments include vaccines that block infection in liver cells, replication of merozoite cells, and or like killing gameocytes. Although promising, those vaccines don't really have guaranteed results. There are also preventative measures like residual spraying, other like biological preventative treatments, and insecticide treated bed nets, but they're usually expensive, and when offered for free by like the World Health Organization, it's usually given to the high risk groups of people first, like pregnant women and children, so other people may not receive such resources. So here we have a graph of malaria cases since 2010. Malaria case incidence went down 27% from 2000 to 2015, so it kept decreasing. But in 2017, the World Health Organization said that the global response reached a crossroads. So malaria was declared eliminated from 6 countries, but 2017 World Malaria Report estimated that most malaria-affected countries were not on track with their reduction targets. So to remedy this, the World Health Organization implemented like a high burden, high impact approach, and it was launched in 2018, targeting the top 10 countries that accounted for 70% of the world's malaria cases. And as we can see, that didn't go so well because there was a huge spike in cases. And in 2020, we see another spike, and people assume that it's linked to COVID because it reduces your immune system, so possibly making it easier to get malaria or to have significant side effects. But research is inconclusive, and we're not too sure about that. So in this line graph, it shows the deaths from malaria since 2010. And while in the other graph we saw a downturn followed by a steep upturn in the number of cases from 2010 to 2020, the number of deaths from malaria has been steadily decreasing. Even though it's a little bit, it's still a steady decrease. From this we can see that the measures to treat malaria after infection have been improving and becoming more and more effective as time continues and as new technologies are discovered. And as prevention of infection has increasingly gotten worse, like since 2015 we saw the spike, but we've developed treatments to prevent death effectively in even high numbers of cases, so there's hope for the future. And here I have my little reference page, and thank you for listening and taking your time out, and that's the end of my presentation.